Good afternoon, everyone. I'm Gina Kardik, and welcome to our last and final webinar from Eurofaang Research Infrastructure think tank. The topic today will be gene editing in farm animal, the science, the application, and the responsible innovation behind this technology. I'm Gina Kardik, and I am from EFAB, and we are the organizing partner for this webinar. Just a few words about EFAB. So EFAB is an organization representing the private sector in animal breeding and reproduction. So as you can see on the right-hand side, our members are breeding organization and reproduction organization who works in different sector and species, which means we have terrestrial livestock, aquaculture, and insect species. And we are based in Brussels because we work a lot in EU policy, regulation, and research. But most importantly, our members from EFAB are also members of FabreTP, which provides the platform for collaborations and research. Because our role is quite clear. We try to bring the practical and commercial and operational realities of the breeding sector in the discussion. So, and this is why today we would like to host this event that brings together both people from the research institutes and the breeding organization to discuss this important topic. Before we start diving into the different presentations, I would like to remind all of you some specific house rules. So please mute yourself when you're not speaking and use the chat if you have any question. If you would like to ask a question, please raise your hand. And I would like to let you know also that this webinar is recorded and will be available on YouTube and Eurofaang's website. So if you don't want to be seen, please keep your camera off. And if there's any problems, you can also write in the chat. I would like to thank Maureen, Joseph Robertson from NMBU, and Emily Clark, who's also helping us behind the scene for this webinar. To begin, I think it's important that we discuss a bit about what is Eurofaang ORI project. And I would like to invite Emily Clark, if she's here, to say a few words, if possible.
Yes, thanks, Gina. So just to give a brief overview about the Eurofaang Research Infrastructure Project. So it's just finished. It finished at the end of December last year. So we ran from 2023 to 2025, and it was really a vision to establish a kind of concept for a consolidated infrastructure to facilitate innovation in genotype phenotype research in farmed animals in Europe. So we had the different partners that you can see on the right. It was coordinated by FBN and then co-coordinated by the University of Edinburgh. And we had a series of, I guess, sort of for Work Package 5 at least, webinars, of which this is one in the series, that have been really helpful, creating dialogue and then feeding into the other work packages, including work packages around industry stakeholder engagement, and then also the use of in vitro cellular systems and then data as well. So it's really been this think tank's really been a foundational part of Eurofaang. And Eurofaang now, although it's finished, it's sort of the foundation for Eurofaang is kind of going forwards with a proposal to the S3 roadmap for research infrastructures called GenoPhoenix, which focuses on linking omics data and phenotyping data. So it's been kind of successful, I guess, and this is a nice way to wrap it up. Does that cover it, Gina?
Thank you very much, Emily. It was perfect. To move on from this, Emily mentioned that the think tank was a foundation also of Eurofaang ORI, but I want to mention why we wanted to do the last webinar today. So the purpose of this webinar is really that we want to bring scientific, societal, and regulatory perspective on gene editing in farmed animal. And we want to have like Eurofaang link project like Geronimo, Rumigen, Gene Switch, who has been, Gene Switch is over, has been delivering results, and Geronimo and Rumigen are finishing in April, also delivering results. And we think it's important for people to hear about it because those projects link those different aspects of gene editing in farm animal. And as you can hear in the recent months and years, there's a growing interest at the EU level, not only because of the work being done in the UK, but also with development in the US and new genomic techniques in general. It really creates this opportunity to kind of have a final and consolidated discussion. And today, what we want is to synthesize all those key scientific advances. So that's why you will see there's people from different countries, different area, EU, non-EU, and the US, to explore those practical applications in breeding system, but also to discuss something that's really important to even start framing the policy behind gene editing, which is responsible innovation, the expectation of society, but also future pathways. And at the end, we would like to identify priorities for the coming years. So in this context, we invited different speakers. And as you can see, we have different sessions. Session 1 would be about the scientific advances in gene editing. Session 2 would be more about the practical applications, where we'll hear about the terrestrial and the aquacultural system. And then session 3 would be about responsible innovation, both in terms of governance, but society and ethics. And then we'll have a nice roundtable discussion with the speakers, but also different people from the breeding sector who would be able to give us their opinion on what's been talked today. So I would like to open the floor now with Simon Lilico, who is from the University of Edinburgh. Simon is an expert in genetic technologies, and he's involved in the Rumigen project, which is one of the projects involved with Eurofaang. He works a lot in the application of genome editing in livestock and has a small focus on disease resistance, but also improving animal models for human disease. He will open this webinar by outlining the science of gene editing in farm animals, where it currently stands right now. Thank you, Simon, for being here today. I will stop sharing so you can go ahead.
Hello. One sec. Talk to myself while I sort this out. That looks about right. And that looks about right. Ta-da! Right. Okay. Hang on. I'm just going to minimize that as well. Go on. Okay. So yes, my name's Simon. I work at Roslin. I do gene editing in livestock, and I'm going to kick off the talk today. The structure of my talk is that I'm going to talk for a few minutes about the tools that we're currently using, and we'll then touch subsequently on some tools that are on the horizon, which could be particularly useful. I'll talk a little bit about the traits that we are trying to modify. I will talk very briefly about some products which are near market, and finally just touch on, are we nearly there yet? How close are we to actually having products on shelves? So thinking about the tools, I don't think for this audience I really need to go into any depth on what CRISPR-Cas9 gene editing is. Obviously, other tools are available, but most of the tools that we're using today to produce products are based on breaking the genome and then reforming the genome afterwards. So I've got CRISPR up here as an example, but as I say, you could equally use zinc fingers or talons in the same space. And once you've broken your genome, you can rely on non-homologous end joining to fix that break, so you get errors introduced and disrupt coding regions of genes. Or you supply a template for homology-directed repair, where you can make very precise changes at your break site. Or you can pair your gene editors up and use them to delete defined regions of genomes. You can also do more subtle things. So rather than breaking the genome, you can fuse Cas9 nicases to a variety of different protein domains, which then perform their function at the binding site. So in the example here, we've got either a cytidine deaminase or an adenine deaminase, which will convert your G to an A or your T to a C. These work reasonably well. They do have some drawbacks, of course. You need to be able to get your guide to bind precisely where you want to make your change. And you have a window in which changes will happen. So anything in this region here, you will convert all of the G's to C's. So it is a more refined tool. You're less likely to get off-target breaks in the genome. but it's also not fully, I think, refined yet for the purposes that we might like to use it for. Along a similar line, you can bind a variety of other things to Cas9 nicases. In this case, for the prime editing, it's reverse transcriptase. In this case, the guide RNA is extended to then include a template sequence on the end of it, which is reverse transcribed to provide a cDNA, which then acts as template for your HDR at the nicked site. So this works for creating modest deletions or insertions at your target site. And some people have used this to reasonably good effect. If we look at the tools sort of historically, when I first came into this finger, we were using zinc fingers. They were great, they worked really well, but they were a real pain to design. When talons came along, I think a lot of groups jumped ship into the talons ahead, simply because it is relatively easy to design and create talons. You can sit down and do it in sort of 2 weeks to a month to get a pair of talons which will target your site of interest. However, it does take that 2 weeks to a month. So again, when CRISPR came along, I don't personally think it's any better than the tools that came before, but it's a darn sight easier. If I had been giving this talk 2 years ago, I would have said I can sit in front of my computer and design guides, and 10 days later they'll arrive in the post. I did this last week and they arrived in 3 days. We have a tool suite available to us in this space for off-the-shelf off-the-shelf Cas9s and guides from a variety of companies, and we can just do all of our design and our cutting. This is a really easy tool set to use to modify genomes. We now have a variety of Cas variants, either from other species or that do slightly different things. They don't have the same breadth of tools or optimization associated with them that Cas9 does. And that's simply because Cas9 was the first. I think these things most certainly will catch up. As I said, we can bind a whole load of different proteins to our Cas, whether it be a Cas9 or other Cases, that will allow us to make changes to DNA at the target site. Alternatively, the things that we can bind can be transcription factors. They can turn genes on and off. They can upregulate. They can downregulate. And with our increased understanding of epigenome and epigenome function with relation to phenotype, there are now options to change the epigenome as well. With respect to the sorts of things that we are trying to change, Alison Vanninanum did a really nice gathering report for EFSA a couple of years ago, which is freely available on the EFSA website. There are a variety of metrics in that, but she was really looking at who is doing what and where. So this is a geographical distribution of first authors on papers that have been published on editing animals for agri-food and feed purposes. There is a reasonable amount of activity in the US, but the vast majority of work's happening over here in China. If we look at the traits that are being modified, I would look at the graph in reverse order. So the vast majority are looking at some form of yield. And the vast majority of this yield, I can tell you, is probably mutations in myostatin, but there'll also be milk yield and wool yield and things like that. The next is reproduction. So this is either looking at making animals that have more offspring, or animals that have no offspring, or animals that have altered sex ratios, for example. Then we have biotic stress. So this is looking predominantly at viruses and bacterial infections and how we can use gene editing to change the genomes of animals to make them less susceptible to disease states. Higher up, we've got allergenicity, so modifying animals so that allergenic proteins that they produce are no longer there. Multiple just means that there were more than one gene edited at one time. Color, I think, is self-evident, and I will not go up to the ones at the top. So there are lots of people all over the world working on these projects. Most of them, I have to say, appear to be in China at this moment in time. We are very good at changing single genes, and multiplexing remains difficult for us to achieve, but it is possible. Colossal Bioscience, in the last couple of years, have published their direwolf, which is not a direwolf, but it is a grey wolf with some phenotypic changes that look like an imagined direwolf. I say all of that sort of tongue in cheek, but this was a wolf that had 14 edits at the same time, right? This is not trivial. This is a leap forward in the science, showing that these things, if not immediately practical for most people, are at least possible. What I really liked was one of the memes that came out at the time. This was in a Scottish satirical magazine called Scottish Eye. And these were the dire Haggy, Carl Haggy, and Haggulus, which were created at the Roslin Institute. And they were obviously prehistoric forebears of the domestic haggis that roams the Highlands today. In the space of new tools that are coming along, one that I think has particular promise at the moment are the bridge-guided RNAs. So just as Cas9 came from bacteria, bridge-guided RNA recombinases similarly come from bacteria. So the picture here looks a little bit complicated, but don't be put off. This is a guide. It is just a far bigger guide than we would have for our Cas9. And rather than one DNA binding domain, it has two. So it has one for the donor DNA and one for the target DNA. And what it does then is recombine the two together at your target site. Now, this was first published a couple of years ago as being utilized in bacteria, but optimization has now been done and remains underway to facilitate use of this in the first instance in human cells. But I see no reason that this couldn't be used in livestock. So, for example, when people give genomics talks and they have their Manhattan plots and they know they have a trait of relevance sitting under a region of that graph, and I speak to them and they say, can you edit that? I'm like, well, how many SNPs are in there? And they say, oh, maybe 500. I'm like, huh. But using this, you could actually recombine that into your genome of interest if we can get it to work slightly more efficiently with respect both to on-target efficacy and reducing off-target effects. But I think this is something that we should certainly keep an eye on as we move in the next few years. With respect to products that are near or turning up near market, ISAAA have a website where they have these things gathered. So these are products that in some jurisdiction of the world have a positive regulatory decision associated with them. And you can search this by species or country or the type of trait or the gene of interest, for example. So if we were to click on species, this would give you a drop-down list of all the vertebrates that have been gene edited for food feed purposes and have approvals associated with them. So if we went down and clicked on the pig, for example, it would say, well, there are 5 positive regulatory decisions in this space at the moment. First was in 2023 in Colombia. Last year, there were 3, US, Dominican Republic, and Argentina, and they were all for CD163. And then you can go across and look at the details associated with that. You can see that the developer is Genus. It was CRISPR-Cas9 used in an embryo as an SDN1 to modify CD163 and produce pigs that are resistant to PRRS. And then you can look at the regulatory documents associated with that. So I say all of this because all of that information is readily available to you should you want to go and look at it. The traits that have positive regulatory discriminations are for heat tolerance. So this is the prolactin receptor mutation in cattle, myostatin mutation in cattle and horses. I'm not sure the horses are intended for food and feed. I think these are polo horses. There is the POLD mutation in cattle and obviously the mutation in CD163 that I just mentioned. Are we nearly there yet? Well, that really depends on who we are and where we are. If you are Japanese, then yes, we are there. You can go to supermarkets in Japan and you can buy gene-edited sea bream, flounder, and pufferfish. So in some limited jurisdictions, products from gene editing of livestock are available as food. My own personal experience thus far is not so much. The closest I've had, John's, who's going to be speaking after me, as you may be aware, he's edited cattle and pigs and goats for the Nanos2 gene, which makes males that are infertile. And what he's shown is that you can take testicular biopsies from a donor animal, culture spermatogonial stem cells, transfer those to a recipient, and reconstitute fertility. And at the Transgenic Animal Research Conference in Tahoe this year, he very kindly produced for us some pork from one of his nanos-edited pigs under Investigational Food Use Authority, and we had some of the finest meatballs I have ever had. So that was really kind. Thank you, John. In reality, too, are we nearly there yet on a more international level? I think the PRRS-resistant pigs are the closest that we have to being there. I think we are nearly there. So, as I said before, we have positive regulatory descriptions in Argentina, Brazil, Colombia, Dominican Republic, and the US. But there are applications in other countries in the world. I would like you to note that the area of the world where we live does not currently have any, and there is probably a very good reason for that. These pigs are only nearly there because they are not being sold yet. And the main reason for that is trade in livestock products is international. So we can have all of the best tools in the world and do all of the best science in the world, but we need to be able to raise a pig in the US and sell products from that pig in China or elsewhere. So we need to have broad approvals for these products. And that is all I was going to say, and I am going to stop there. Thank you very much.
Thanks a lot, Simon. We won't take questions right now for Simon because we will have a discussion round later on. I would like to give the floor now to John Oatley, who is joining us today from the other side of the world, and it's really early. But John is an associated dean for research and professor of molecular biology, specializing in germ cell and animal genome engineering. His work bridges the fundamental research, but also real-world breeding application in terrestrial livestock. Thank you for being here today, John.
Yeah, thank you for the introduction, and thanks to Simon for setting the stage. I can go quickly over some of the things that I was going to address as well. So I'm going to focus on how integrating gene editing into livestock production with a couple of examples, and also sharing some of the experiences that I have had with navigating regulatory landscapes, but also interfacing with producers and consumers to try to get buy-in for the things that we're trying to develop as traits across various livestock. So at Washington State University, I direct a research program that we refer to as the Resilient Livestock Program. We primarily use CRISPR-Cas9 genome editing to do a variety of trait development in the major food animal species, including swine, small ruminants, and cattle. We work on a variety of things, trying to improve the welfare of animals, conferring disease resistance, developing reproductive capacity, and also working on altering the properties of skin. I was also a member of a National Academies of Science in the United States, a committee that was tasked by the US Congress to develop a scoping report for heritable genetic modifications in food animals. And we published that report early in 2025. If you're interested in the report, if you haven't been able to get access to it, you can contact me by email and I can share it as a PDF document. So as most of you on this meeting know, selective breeding is a tool that's been used for centuries to try to drive genetic gain in livestock populations. And although powerful, it can be messy and slow to progress. One of the reasons is that it's based primarily on observable phenotype. It can often take 5+ generations or 10 or more years to make incremental changes in the traits of a population. And it can also lead to inadvertent selection of negative phenotypes. One of the prime examples that I like to use to portray that is this bull Rito 9J9, probably the most used artificial insemination sire for the Angus breed of cattle, and was selected for all the great properties that he had in growth, performance, and meat quality, but was also a recessive carrier for curly calf syndrome, which took decades to breed out of the Angus population. And so those limitations of selective breeding were the basis for the development of marker-assisted selective breeding. As many of you know, now we have all these SNP arrays that are looking for polymorphisms in the genome that are linked to a variety of traits that can be selected for, such as marbling, disease resistance, average daily gain, fertility, breed identification, etc. And the real purpose of marker-assisted selection is to increase the rate of genetic gain to drive trait development. But marker-assisted selection also has limitations. And that includes that a lot of times the SNP is an association with a trait, and that doesn't mean that it's always a driver of a trait. Also, it does not mitigate the inadvertent selection of negative phenotypes, and oftentimes requires crossbreeding, which is dilutive of the base genetics. And so the real promise of precision breeding using gene editing to develop traits is to utilize molecular tools, state-of-the-art molecular tools that Simon just nicely covered, and combine that with the loads of DNA information that we now have for various breeds and populations of livestock to get efficient, precise genetic improvements, and do this in just 1 to 2 generations and mitigate the potential dilutive or drift genetics that can happen and remove the possibility of negative selection. So most of the developers in this space are working with CRISPR-Cas9 as their gene editing tool. I won't go over how it works because Simon just did that really nicely. And so one of the powers of CRISPRs is that it can write any of these SNPs that people are currently selecting for with marker-assisted selection. These SNPs can be written into the genome of animals to get genetic marker integration without crossbreeding and really engineer precision breeding stock. Another power of CRISPRs is that it can write novel genes. And so we can generate new genotypes that are designed to solve major production challenges or issues. And the prime example, as Simon just mentioned, is the PRRS-resistant swine through modification of the CD163 gene, which they're able to generate pigs that are resistant to the PRRS virus infection. And this has now been approved in several countries, including the United States, with an FDA approval decision. And so gene editing really can impact across the production cycles of livestock. I'm just going to use beef cattle here as the example, where there's several spots in the production cycle where there's opportunities to impact traits and therefore improve efficiency and address food security. And so in the beef cattle production cycle at the cow-calf stage, there's opportunity to improve reproduction. There's opportunity to improve udder health, address some of the major welfare considerations like that are production practices such as dehorning. Also the opportunity to engineer disease resistance, including a variety of the major diseases that impact beef cattle. When animals are moving into the stocker or backgrounder and finishing phase, there's additional opportunity to engineer traits for disease resistance. opportunity to address intestinal parasites, which will help with average daily gain in the feed conversion ratio, opportunities to develop alternatives to sterilization, to improve feed efficiency, and also opportunities to reduce methane emission in the production animals. And so in trying to develop traits in a production system like beef cattle, really it starts at the cow-calf stage. And so what that means is that we have to apply gene edits into developing a breeding stock. And so there's a couple of major ways that developers are currently doing that, with the intent that we're not just making prototype animals to test a phenotype, but also the possibility of creating breeding stock. And they all involve editing of embryos or generation of edited embryos. So one of the strategies is to use in vitro production of embryos through in vitro fertilization and embryo culture at the zygote stage just after fertilization. An alternative is to use somatic cell nuclear transfer, where a fibroblast nuclear genome is engineered and then used in an enucleated egg to essentially generate a zygote stage embryo. And so this is where the gene edits are applied, various strategies to introduce CRISPRs and other gene editing strategies into the early embryo, transfer those embryos, establish a pregnancy, and hopefully generate founder animals that have the designed traits. So as I said, this is an approach to generate prototype animals to determine whether the gene edits that one is hypothesizing produce the intended trait, and these will be prototype animals, or to develop potentially breeding stock that can be used to integrate those edits and develop those traits in populations. So one of the positives of this type of strategy is that really any sire, any dam can be used for zygote generation, so you can get breed specificity and even population specificity. A negative is that oftentimes, especially with direct embryo editing, the zygote stage, it almost always leads to some level of mosaicism, and this means that there's unknown heritability in the trait, or unknown heritability of transmission of the genetics for trait development. And in the case of somatic cell nuclear transfer, there's very low efficiency and developmental abnormalities that form. So there are some limitations of this strategy. And so really a key element in developing gene editing for trait development in livestock is to be able to get widespread dissemination of those trait driving genetics into production populations. Just making one animal is a major feat, but being able to get the genetics carried by that animal into population levels for impacting production is a whole nother challenge. And so that means that we need to amplify the availability of gametes that possess edited genomes. And this means reproductive technologies are very important. And one of the reproductive technologies that can be used is artificial insemination. This is something that was commercialized over 60 years ago, has been extensively used in dairy cattle production to improve the genetics behind milk production. And quite from a simple standpoint, we can have a desirable, elite, high genetic merit, whatever you want to call it, sire somewhere in the world, collect his sperm, put it in a cryopreserved state, and ship it around the world for artificial deposit in the reproductive tracts of females. And so while a very powerful tool, it does have its limitations. In small ruminants, it's a technical challenge to apply. Artificial insemination typically requires a surgical procedure. Only about 1% of small ruminants globally are bred by artificial insemination. In beef cattle production, it's a logistical challenge in that the animals must be tracked, females must be tracked for their estrus cycle or highly manipulated with exogenous hormone injections in order to get artificial insemination or deposit of semen at the right stage of the estrus cycle. And because of those logistical challenges, only about 7% of beef cattle globally are bred by artificial insemination. In swine, inefficiencies in the ability to freeze sperm is a major limitation for global distribution for use in artificial insemination. And so for many years, we've been working on a solution to this that we call surrogate sires, where we can generate males that are going to produce sperm with trait driving DNA that may not be their DNA. And this can be disseminated by natural mating, essentially taking all the advantages of artificial insemination for genetic gain, but applying it in a natural mating setting. So this is a CRISPR-Cas9 application for this project that we have been working across all the major livestock species to try to develop as a breeding tool for efficient propagation and dissemination of trait driving or even edited genetics in livestock populations. And the basis of this technology is that spermatogenesis, the process of sperm formation in the testicles, is a stem cell dependent process. And so the surrogate sire's breeding concept looks something like this, starting with a elite high genetic merit or gene edited sire. We can isolate stem cells from pieces of testicular tissue, transfer those stem cells into the empty testicles of surrogate males, a battery of males in which donor-derived sperm production happens, and they can breed by natural mating to generate progeny with the donor genetics rather than their endogenous genetics. And so the key here is that we have to be able to eliminate the endogenous germline in the recipient sire so they don't make their own sperm, and all the sperm they're making carry the genetics of the elite or gene edited sire. And so this is where the CRISPR intervention came into play. So we've been able to do this at Washington State University with a number of collaborators in swine, beef cattle, and small ruminants through knockout of a gene called NANOS2 that makes the males sterile, but their testicles are otherwise able to harbor the engraftment of donor sperm producing stem cells and generate donor-derived sperm. So we're now working to develop surrogate sires as a breeding tool for impacting global livestock production, not just in North and South America, but also the UK, Africa, India, and China. And so surrogate sires has another utility that we believe can be used to enhance the precision of gene editing in livestock, and that's to overcome these limitations I was talking about with direct embryo editing. So in direct zygote embryo editing, adding our CRISPR-Cas9 components plus or minus a repair template, oftentimes mosaicism happens. You have to genotype the animals postnatally before you know what you have, and this leads to unknown heritability of the edits. In somatic cell nuclear transfer, although it's known what the edits are going to be prior to transfer, it has low efficiency and developmental abnormalities, thereby limiting it as a tool to be able to generate breeding stock. So we believe that surrogate sires concept can also be used for direct germline editing, getting the edits into cells that are directly going to produce gametes and already knowing what's going to be heritable before we even derive the gametes. So that looks something like this, collecting stem cells from testicular tissue of a donor male and introducing CRISPR-Cas9 or some other gene editing strategy into cultures of sperm producing stem cells, selecting those that have the edits that we want, doing clonal expansion where we can screen for a variety of things, followed by transferring those into the testicles of surrogate recipients. And now we have a natural breeding setting where we can generate progeny with the donor genetics plus the gene edits that we want. So we've been working on this for a bit and we're getting closer every day. And we think when fully developed, it can address a number of limitations, including allowing us to determine both intended and unintended edits before we even derive gametes that can be transmit heritable information. We can test the durability of the edits, we can predict heritability, and we can stack multiple edits all in vitro before we even generate gametes that are going to transmit that information. So I'm just going to finish up by talking a little bit about beyond the science and the work that we've done in trying to advance genome editing applications from the lab to livestock producers. There's a couple of challenges. One is public acceptance of the technology in modifying their food, and the other is regulatory policies. There's still a GMO stigma, and even though the science and the intent of gene editing is very different than conventional or traditional GMO, it's still got a stigma with the public. And so there's a need for a new narrative, developing a new narrative with the public around the science and the purpose of the science. I think this really involves community engagement as well as developing better strategies for science communications. On the regulatory side, there's still antiquated and misaligned regulatory landscapes in a lot of countries to the current state of the science. And this requires some level of modernization, streamlining, and becoming more cost effective. Harmonization across the US Department of Agriculture, the Food and Drug Administration, and DEFRA in the UK, I think is critical in achieving this. One of the biggest things that I've spent the last couple of years outside of the lab working on is developing better stakeholder engagement and collaboration. I've started working closely with the National Pork Producers Council in the United States, the National Cattlemen's Beef Association in the United States, and the American Association of Veterinary Medical Colleges to develop collaborations to improve both producer and consumer understanding of the science and technology. I've also been working with the Food and Drug Administration, U.S. Department of Agriculture, to improve how we develop and regulate gene editing in livestock. And the last place where we've started to work is trying to influence what are the future farmers of America. This is the FFA in the United States, but I think lots of countries have this types of associations. So young kids that are developing an interest in farming and developing knowledge base for those kids as they develop as the future farmers and ranchers. And so through all this engagement, I've realized there's a few overarching concerns with gene editing in food animals. One is animal welfare. Regulatory agencies and the public are concerned about developing unanticipated negative phenotypes through gene editing. We're worried about pathogen gain of function. Are we going to drive the evolution of new viruses? There's concerns on societal safety. What's going to happen if we alter the composition of meat and milk? Will we derive novel allergens in the food that the human diet hasn't seen? And are we going to increase greenhouse gas emissions from our livestock and therefore have a negative impact on the environment? And then there's producer adoption of the edits and the traits that we're trying to develop. When I speak with producers in the United States, one of their biggest concerns is consumer acceptance. They're worried if they develop it as part of their breeding strategies and the consumers won't eat it, then it's going to destroy their business. They're also worried about trade impact. What happens if we now open up or close down new trade channels that are going to impact their ability to be profitable? And the last is access to genetics. They're really concerned that just a few companies are going to have the developed genetics through gene editing, and they're going to limit access to only some producers. And so I think this is also really important for producer engagement. And so again, this National Academies of Science Committee on Heritable Genetic Modifications of Food Animals is trying to address many of these things that seem to be common concerns for the producer, regulatory agencies, and the public. And so just a summary of the findings from that report, that committee focused on the fact that phenotype is the priority consideration. You can engineer any sort of genotype, but if it doesn't have your expected phenotype, then it's not really going forward as something that will be integrated into production. Heritability is key. Not all gene edits that can be detected in somatic cells are going to be heritable, especially when dealing with mosaicism. There's a need for comprehensive reference genomes to be developed. The committee found that largely across livestock populations, the reference genomes were not developed deep enough to be able to fully understand the impact of gene edits and potential unintended edits. There was concern about creating abnormal or toxic composition of meat and milk, but the committee found that this is unlikely to get outside the range of normal for the composition of meat and milk. It was almost impossible to create some sort of toxic composition that didn't also kill the animal. The committee found that food from livestock with genome edits is generally safe to consume. There is no empirical evidence to suggest that there would be any harm to the consumer. But the committee did recognize that there is potential to drive viral evolution by engineering disease resistance and considered this a moderate risk, but not a risk that's much higher than vaccinology that's trying to develop the same thing. And so one of the last things I'll touch on is, will consumers accept products from gene-edited livestock? My experience is FDA approval for CRISPR-edited swine. United States, we got an investigational food use authorization to harvest a group of pigs carrying Nanos 2 gene edits. And we're able to produce that and enter it into the human food supply. This was a couple of years ago now. And I'll tell you, we've had no pushback from the public in providing this. The public reaction was that we sold out of 200 pounds of the Nanos 2 CRISPR-edited sausage in 1 month. There was very little voiced concern about consuming this product. And now we're working with the FDA to try to develop Nanos 2 CRISPR-edited beef, and hopefully we're going to get some sort of investigational food use authorization for that soon. And so just to wrap this up, what I hope I've been able to show you is that integration of gene editing in livestock production is a modern tool for diverse and rapid trait development that can generate impact across production cycles. The heritability of the trait-driving gene edits are key. Innovation in reproductive biotechnologies is needed to amplify and provide widespread dissemination of edited genetics. There's a need for new narratives on genetic engineering with both the public and the producers. And modernization, harmonization of global regulatory systems is essential to be able to get this from the lab to the farm to the table. And lastly, producer and consumer acceptance, at least in my experience, is largely positive. And this is something that we're working to smooth that road and develop that new narrative so that we can overcome what could be a bottlenecking or roadblocking limitation to getting the science into impact. So that I'll say thank you. This is my email address. If there are questions that I can't answer right now, please feel free to contact me and I'd be happy to try to address them. Thank you.
Thanks a lot, John. John will have to leave due to some lecturing responsibilities. If you have 1 or 2 questions, we can ask John before he leaves in the audience, or if you can also write it in the chat, that would not be a problem. Any questions?
Hello?
Yep, we can hear you.
I'm sorry, my camera is not working, but I guess you hear my voice.
Yes, we do.
Yeah, I would like to thank the two speakers, but I have a concern relative to Dr. John's presentation. Please help me to understand. You are saying that classical genetics or animal breeding is very slow and messy, and the alternative is go through gene editing. I would like to be convinced on that by recalling that animal breeding, its concern is not the animal itself. Its concern is the variability within the breed. This raw material is a safe pathway for the present and for the future, because you have always alternatives and you can adapt and change. This is compared to what you are saying, editing genes will be the solution. I hope that my question is clear. Thank you.
Yeah, I think I understand your question and I'll try to address it now. I think we could probably spend an hour debating this. So I'll try to do it really quickly, but certainly I didn't mean to discourage or downplay selective breeding. It's a tool that's been used for a very long time and has made great gains, but it is slow. It does take many generations to be able to even make incremental advances in traits. It's not something that's going to change a population or 1 or 2 generations dramatically. And so it is relatively slow compared to the speed at which we can use CRISPRs to precisely write in some sort of sequence or change a sequence that is going to immediately drive a phenotype. With selective breeding, there's also, as the example I showed, the possibility of inadvertent selection of a disease-causing genotype, which may not manifest for many years, because if it's recessive, it's going to require a lot of backbreeding and crossbreeding to get back to creating some sort of disease state. And so that is something that can be mitigated with gene editing. In terms of safety, we who are developers of gene editing believe that it's safe. We're not going to be generating genotypes that are going to pose any sort of risk. When you think about developing a trait, if we're not getting the expected phenotype and we're getting some unexpected phenotype through the research and development phase, it's not going to go forward into a production setting. Anything that's going to pose a risk to the production animal or to potentially consumer safety is going to stop very early in the research and development phase. And so the things that actually make it into producers' hands or into the market is going to be fully vetted before it even gets there. And so I think that it's a combination of traditional selective breeding, marker-assisted selection, gene editing, All of these things need to come together to really impact food animal production to address the future of food security. So I'm not saying they're going to replace gene editing is going to fully replace selective breeding or marker assisted selection. In fact, I think they complement each other, and it's just going to be what producers and developers want to use as their tool to be able to get the traits developed in the way that they want for their production systems.
Thank you. I was reminded to present myself. I am Dr. Jamali. I graduated from Iowa State University, and I am an animal breeder. And I have been in the field for more than 40 years. And I value the animal breeding science as a tool to improve productivity under different environmental conditions. As a matter of fact, being slow is not a bad behavior. Being slow will allow you to adjust whenever you need it. Thank you.
Thank you very much. I will give the last question to Mathieu, who raised his hand because John has to leave for his lecture soon.
Yes, hello. My name is Mathieu Sommers. I'm European veterinary specialist in laboratory animal medicine, working at Wageningen University in research. And as you probably are aware of, in Europe, there are, as I understood, much more restrictions to the use of gene editing in animals, definitely not for production, that is currently forbidden. However, my previous job was in a biomedical facility where gene editing of rodents is quite common, especially mice. My question is if people in this lecture are interested to get more in-depth knowledge, could you or John or a combination of experts in this meeting come up with a short list of literature where we could read up in understanding the possibilities of gene editing in livestock, not only for these, let's say, production-oriented translational purposes, but also for more fundamental research questions where we try to discover mechanisms, etc., which we also work a lot on at the Wageningen University. Is that something that you would be open for to feed to the same email recipients? Reading a short reading list that could help us to get up to speed with what you are doing and all the possibilities in livestock. Thank you.
Yeah, I appreciate that comment. And I think that's something that we should try to do. Oftentimes we create these massive reports. And I'll just take the National Academy of Sciences report that we generated. I forget, it's probably something like 200 pages long, which is really a tough read. And even though it has a lot of information about what's being developed, the potential concerns, comparison to just conventional selective breeding as well, it's very difficult to read through the whole thing and pull out the key points, even though there is a summary. So I think that that is something that we as developers of gene editing, gene edited trait development in livestock, could try to put together something that's an easier digest for just people looking to just get general understanding of the state of the science, what's being developed. I think sometimes we go too far in the stuff that we generate and try to make it too big. So absolutely, I appreciate that comment, and I'm happy to engage with others in the field, people that are other speakers in this session to try to do that. Absolutely appreciate that.
Yeah, because
Thank you,
in
John.
mice I was heavily involved in gene editing, and there it is quite straightforward. And we made gene edited mice strains all the time. But I think livestock is definitely behind that, and I'm not fully aware how much behind it and what are the let's say the struggles that we have to anticipate if we want to use gene editing for, let's say, more fundamentally oriented research questions, which would be very helpful technique to add to our portfolio.
Absolutely. One of the biggest differences, of course, between rodents and livestock is the generation interval. Taking
Yeah.
much longer to get from a gene edit to a discernible phenotype. And this mosaicism thing that I talked about is a real issue because I also work in rodents, and anytime we have animals that are mosaic, we just breed it out, right? And we segregate out the mosaicism really quickly. Well, when you think about livestock, especially cattle, that's years, not months, to be able to do that. So that's one of the limitations that is posed with livestock. But yeah, absolutely. I think we could go on discussing this for a while. But yeah, I'm happy to try to develop something that's going to be a more, no pun intended, but palatable understanding of the state of the science in livestock, not just for agriculture, but also for biomedical modeling.
Yeah. Thanks a lot for this very interesting
Thank
presentation.
you very much, John. One last, I think it's a yes/no question. So we get a question in the chat from Christian Luft from the University of Neubrandenburg, and he's asking if you believe that the use of these technologies could lead to the formation of monopolistic structures in the breeding sector.
I think if not developed correctly, yes. But I think we look at the way that other breeding strategies are applied, assisted reproductive technologies, as an example, they have that potential as well. So I think we just need to have a smarter design to the way that we're developing the genetics and then making access to the genetics. And being at a land grant university in the United States, right, I'm not about trying to put roadblocks or limitations into access to genetics. It's more providing things that would be available to producers. However, we get that into producers' hands, I don't know, and making sure that we don't bottleneck the genetics is going to be really important. And so it just takes purposeful design to the way that we're developing and disseminating the genetics.
Thanks a lot, John. We really appreciate it, and we appreciate your time, and we hope we can stay connected through the think tank and other work of Eurofaang and gene editing purposes. Thanks a lot. Have a nice day.
Thank you.
I would like now to invite Anna Vagelus to present. I will be sharing my screen, but Anna is a leading researcher in functional genomics and reproductive biology in Atlantic salmon. Her work uses gene editing to develop sterility models, improve disease research tools, and address key sustainability challenges in aquaculture, including unwanted maturation and interaction with wild population.
Well, thank you very much for being invited. And also thank you to the two previous speakers. So I'm supposed to tell about the fish field in this area. So I think we got a lot of introduction from the first two nice talks. But I will tell a little bit about the practical aspects then of editing and breeding in aquaculture. And maybe just give a brief background first to the aquaculture field, and then how you edit in aquaculture, and then give a few examples of phenotypes that are of interest in aquaculture. So my group has been working for about 14 years doing types of edits in salmon to try to understand the biology of the salmon, but we have also developed lines which might be interested for production purposes. So when you go back and look at aquaculture, it's a very much growing industry, and the production of aquaculture species has in last year then surpassed the production of wild fisheries, and there's a vast amount of different fish species. There are 27,000 fish species in total, and of course a few of these are in aquaculture, but it even though becomes quite complex when you work with aquaculture, because all of these have of course different life histories and also very different genomes, but there are a few things that are in common when it comes to editing that I come back to. So I just wanted to show the most famous transgenic fish starting here, which is the most famous transgenic animals. I think so far it's the aqua bounty fish, and this was then created by traditional genetic engineering by putting in the growth hormone in a random place of the genome. And then of course this editing technologies, which were very nicely presented by the previous presentation, have then moved from this unprecision to this precision, and this type of changes are then very similar to those occurring naturally. And we have really a few fish species that are already produced in a commercial space, like this red sea bream, which you can buy at the market in Japan, which has the limitation that causes them to have double muscles. This is the Belgian blue gene or the MST1 gene. But then how does the gene editing differ then from farmed fish, from farmed animals on land? Well, of course, you have them, that they are produced, the eggs are, and the fertilization is occurring outside the animal body, which is, of course, making it easier to do the editing. Also, there's a very, very high fecundity. Typically, a salmon female in production can produce up to 10,000 eggs. So this is, of course, a good thing for breeding if you want to upscale your edits. But there's also drawbacks with fish because they also have duplicated genomes in history. So there are often many copies of each gene. So in salmon, for example, we can have to 4 paralogs of each gene. This can be both good and bad for editing. If you want to reduce gene expression, maybe only not to remove the gene, it's nice to have paralogs. But often, if you remove one paralog, the other one take over. So maybe sometimes you have to knock out or change all 4 paralogs to get the phenotype of desired. And then, of course, many of these species are reared in open. They're water living and they're reared in open cages in the water. So SKP is a much larger problem here as opposed to land living farm animals. And also another thing which is complicating is the long generation times. For example, the salmon that we work with have a generation time which is 3 to 4 years, which then takes a very long time if you want to move to F2 generation. I'm coming back to that. So the generic method for introduction of edits is easy, of course. You can inject it in the first cell stage of embryos. They're outside the body. So this is the most common way, and most of this CRISPR edits are quite easy. You get a very, very high efficiency already in the F0 here, illustrated by knocking out the albino gene. And there's 100% mutation rate in many of the embryos, so it's very efficient. So we can study the phenotypes in the F0, but you can also have other species which are not so easy to inject. For example, we work with flatfish also, which are the eggs are very difficult to inject. Then you can use methods like sperm electroporation, and also I'm going to come back to that, cell lines or surrogacy. So what we have done to be able to do our science, to not wait forever, is that we make the crispant. We study the crispants too, if they're very highly mutated, but we also have jumped the step when it comes to getting the knockouts. And we have crossed that crispants to save 3 to 4 years of science. But of course, this is not the case when you want to produce a line for breeding. So we are also doing crosses where we cross out the classical, where you cross out the crispant to a wild type male, for example, and then you wait for the heterozygous and cross out the heterozygous. But this is a way that we have been doing many of our research trials to save time. So when it comes to the typical phenotypes that are studied in fish, there's this that has to do with growth, which is mostly this double muscle, but here you have several fish species which are already in production. Then reproduction is a trait that's very popular because you want to have sterile fish in the facility. It's common to study them in terms of sex determination. You want to maybe make all male, all female population, which can be good for predictable production. And then so far, it hasn't been many studies on disease resistance, but this is, of course, of high relevance. And then also it comes to meat quality. That's also something that's done some science on. So another thing that is special for fish is that vaccines work very well in fish, but the cell lines to produce this are very scarce. So these technologies can be used then to make better cell lines. But also, it's very relevant to use the technology maybe for them germ cell cultivation and then editing. And as it was already shown in the previous presentation, surrogate broodstock technology has been existing in fish for for quite some time, 20 years. But now with the editing, it allows the combination of both maybe having superior breeds that you just bring out the germ cells and then put into sterile host, and maybe these sterile hosts are then produced by editing developmental genes like dead end or PV, but like we have done, or that you can bring out these germ cells and do the editing in vitro. This is not yet done, but it will be in the future, I think, and we have work on this. So, but of course, I'm going to give an example here because edit for disease resistant is very relevant, and also I think it's relevant for the previous questions after the previous talk, because one of the biggest problems in salmon aquaculture is the salmon lice, and there have been big efforts to try to breed for this resistance to salmon lice, but it hasn't working, even though companies have been working on this for a long time. So then we are involved in a project where we try to translate the resistance which is occurring to this lice in another species, the coho salmon. So now we are then having a set of candidate genes from coho salmon that we're trying to put into Atlantic salmon so that we can cut and paste and then hopefully get a salmon that has resistant to this big problem in Atlantic salmon. And then my group has focused a lot to work on sterile salmon, both to inhibit germ cell formation and prevent maturation. This is to be able then to protect wild strains if they escape, but also to prevent from the welfare consequences of earlier maturation in sea cages, because that's not so good. That's increasing the disease susceptibility. So we are currently working on several methods for sterility, but one of the ones that are really cool, I think, is this surrogate methodology, in which we have developed 2 edited lines, which we are now on the F1 generation for FSH receptor, but for the dead end knockout, which is then this germ cell-free surrogate host, we are now on the 3rd generation, and then we can produce by crossing out heterozygous dead end parents, then create dead end knockout fish that can be used as surrogates for this FSH or germ cell. So FSH receptor is sitting on the supporting cells of the germ cells, and when the fish decides to mature in response to FSH, they cannot mature. So these fish which are knockout for FSH receptor, they are what we call forever young. But by taking out this germ cell and put them into this dead end background, we can then make them fertile broodstock, and these fertile broodstocks will produce only FSH or minus offspring that will never mature. So this is an option to get to large-scale production of sterile salmon. We are currently having several lines which we have tried out to put these germ cells in, but we have not yet succeeded to produce the germ cells in this particular genotype. However, it has been done in Medoka, and also colleagues of mine have just been able to show that we can do surrogacy in Atlantic salmon. So I think with this concerted knowledge and the lines we have, I'm sure we can produce sterile salmon at large scale soon with this technology. So that was all I had to say, and in the end I just wanted to thank my group and the collaborators of these projects. Thank you.
Thank you very much, Anna. So no questions. Anna, thank you very much for your time and for your presentation. We will make Anna's presentation available later. I will now like to go towards the 3rd part of our session and give the floor to Andy Greenfield. So Andy is internationally recognized actually for his expertise in ethics and governance aspect of emerging life science technology. But he's also the external ethic advisor to the Rumogen project, supporting responsible innovation in livestock gene editing. Thank you, Andy, for finding the time today. I'll give you the floor.
Thank you. I hope you can hear me and see that slide. So
Perfectly.
I've entitled this Reflections on the Ethics of Genome Editing and Farmed Animal Breeding. Just some disclosures, declarations. These are all my opinions and not necessarily anybody else's. So John mentioned long, complicated reports. And what I'm going to say is really prompted by looking back at this publication. This is now almost 5 years old, a publication from the UK's Nuffield Council on Bioethics, which was an exploration of genome editing and farmed animal breeding, social and ethical issues. And I was a member of the working group, as were a number of familiar names. I'm sure you'll be familiar with some of them. It's rather a daunting challenge to have some kind of systematic attempt to understand ethics and farmed animal breeding, primarily because it simultaneously engages three areas of our lives, aspects of our lives that are incredibly important to us. So our interactions with non-human animals, what are our obligations to animals? How do we interact with them in different contexts as well, be they companion animals, research animals, wild animals, farmed animals, and so on. Focusing on genome editing raises the whole topic of biotechnologies and technologies in our society. How are emerging technologies integrated into our societies? Who decides? How are they regulated? And then finally, of course, farming itself and food production. And food is an incredibly important aspect of all of our lives. And so when you have to engage a topic such as this, where three of these have to be considered simultaneously, it's very, very challenging. So it took about 18 months to do this, although there was a pandemic to navigate as well. There was a diverse working group, not just academically diverse, but also in terms of diverse commitments. So people like me, which again, a declaration, I am amenable to farmed animal genome editing, and I come out of the biomedical research space. So I was a mouse geneticist who used genome editing in mouse genetics. So I was amenable. There were others who were much less amenable. So it was a really complicated set of conversations. It's a wide-ranging report, 200 pages. It's international in scope. It adopts this what we call a critical approach. It doesn't ask the question, "I've got this shiny new technology. What can I be allowed to do with it?" Rather, it says, "What are the challenges that exist? And what are the different ways that we can address those challenges? And what role is there for a technology such as genome editing in addressing those challenges?" It is undoubtedly the case that the report wouldn't have been written by any of the individual members of the working group. It's a compromise. And it's very difficult to summarize. And of course, when I do attempt to summarize it, my choices are themselves value-based. So, so that's again another declaration. What types of objection to genome editing in farmed animals did I experience in my time on that panel? And we obviously interviewed many, many stakeholders. There were some fundamental, what I call here fundamental objections to the idea of farming of animals, objection to the instrumentalization of animals and their lives. As I say, we did discuss this topic with people who would rather not farm animals full stop. There were those who objected to meat eating for a variety of reasons. Some objected to the speed of technological intervention. And of course, we've already had that conversation today about slow versus fast. And that often arose in the context of a concern about naturalness, about which much could be said, but I may come back to naturalness. There are environmental concerns. Of course, there are proposed uses of genome editing in animals which will mitigate climate change, but there are still some individuals who are concerned about the sheer number of animals that we have on the planet that are being bred for consumption. There's a lot of concern about commercial interests versus public interests. Remember, I'm talking to you from a UK stroke European context. So, contexts across the world, culturally, politically, are diverse and they will be different. And certainly in this context, there was expression of concern about commercial control. There were also concerns about the exacerbation of the impact of intensive farming. Some of the proposed changes may allow animals to endure intensive farming, when in fact what we need to do actually is to change the methods of intensive farming. And there are of course concerns about the whole future of farming. So these were all common expressions of concern by some of the expert stakeholders. Some of the key themes of the report, it identifies some challenges to the food and farming system. I have to say it's rather a gloomy, depressing read at times in terms of the assessment it makes of the global farming and food system. There are challenges for us in terms of animal health and welfare, of course, also human health, zoonotic diseases and etc. The sustainability of the food and farming system, its impact on the environment, and so on. So it identifies those, and then its basic ethical approach is to just talk about what it describes as basic interests. So all animals, including humans, have basic interests. They must be met, and that is part of what it is to ensure justice. So the production of food and feed should be achieved in a way that secures the basic interests of humans and animals. New technologies, therefore, when they're introduced into this food and farming ecosystem, should aim to secure and promote basic interests. Now, genome editing might be able to do that, but there's obviously a potential for it to be put to uses that do not respect animals' basic interests. So you could say that the theme is that this is potentially a dual-use technology, that it can be used to do good, it could be used to do harm. And it also makes it clear that public engagement is key to ensuring that we align policy with public interests. There was a public engagement exercise as part of the project. And some of the themes that arose from that, I think, were very interesting. And I'm sure Phil, who's speaking next, will address perhaps some of these. There was interesting observation that people responded in a way where their identity was almost to some extent constructed by the process of engagement. So they are either approached as consumers and asked questions about what they considered to be desirable in terms of food production and the kind of food they want to consume. But they were also asked to consider these questions in terms of being citizens rather than just simply being consumers. And attitudes can change depending on the identity that is adopted. When acting as consumers, they obviously still wanted safety of their products, but they also wanted choice. As citizens, they tend to be more concerned with animal welfare and also justice. So, what kind of food system is best in the long term? What does sustainability look like in the long term? The actual technical details of breeding technology seem to be relatively unimportant to them. It didn't seem to matter to them, whether we were using base editing, prime editing, or traditional CRISPR-Cas9 or TALENs or whatever. It was the actual intention of the application and the intention of the breeding that was more important to them. And there were questions such as, who benefits? Is it the producers or the consumers? And of course, it was more acceptable if it was the consumers that were benefiting rather than the producers. And what are the impacts likely to be on animal welfare? And again, will intensive farming practices be entrenched? There was also expressions of concern about naturalness and the environment. Now, there's been a lot of work on the concept of naturalness. I think for scientists, naturalness is a term that we use empirically. It's just what happens in nature, some of which actually is good for humans, much of which, of course, is very bad for humans. I think it's used in a slightly different sense when publics are engaged in these kind of exercises. They're using it in a more normative sense. It's about, it's about questions of contamination of scientists and potential their hubris in interacting in ways that they don't understand. So there's a complex use of that word. One question that emerged from this engagement exercise is, whether or not applying genome editing technology will take us towards or further away from the kind of future agricultural systems that we should be aiming for. So, as I say, if I attempt a very, very, very short summary of the report, kind of a summary even of the executive summary, it's very difficult to do. But it would be something like genome editing, or indeed any other breeding technology, will always be introduced into a particular social and political context. And the one that we're discussing here, the global food and farming system, is one that, in the opinion of the report, is currently unjust and unsustainable. So it's into that context that it will be introduced, and that has certain consequences for how we consider the role that could be played by genome editing. And as has already been mentioned earlier, it will have consequences for the kind of narratives that were likely to be accepted or not. We need, according to the report, reform of governance and regulatory systems. New frameworks of government and oversight are required, particularly oversight mechanisms that do promote and secure justice for a food and farming system. But of course, discussing regulation raises all sorts of issues of proportionality, what degree of precaution is required, the importance of innovation, safety assurance, opportunity costs, the costs of not adopting new technologies, what is in the public interest, which of course I remind you is not the same as what the public are interested in. It is what is in the public interest, accountability, trustworthiness, trust. There are so many different elements to this that it's a very, very complex story. If I had some criticisms or comments to make about the report, I might say that why wasn't it simply called farmed animal breeding, social and ethical issues? I mean, to some extent, when reading the report, it seems, looking back, that genome editing plays a relatively minor role. And indeed, it's very technology neutral. It really is focused on, what are the intentions of breeders in generating the animals that they generate, and what are the potential benefits of that breeding for the animal and for humans, and the impact of all that will have on the system that we currently have. And so, to some extent, people who wanted to read a report about genome editing and what it can do will be, to some extent, a little disappointed. But it's interesting also that the fact that genome editing is used as a vehicle to explore the global farming system can inadvertently perhaps promote technological exceptionalism, by which I mean this idea that we always seem to focus on new technologies rather than the everyday impact of everyday policies, which don't necessarily require technology at all for their running. We can think about all sorts of policy initiatives that don't involve technology that would have profound impacts on the food system, such as, for example, policies to eliminate waste. So why do we always seem to focus on technologies is sometimes, I think, a concern. Finally, we do need to have a debate where we are transparent about our commitments. And one question is whether or not justice for all as a mantra can really ultimately avoid concluding that any instrumentalization of animals is unacceptable. And I think that what we mean by justice is something which is itself very complicated and one that's going to need to be worked through very carefully. So where does this leave us? I think the case can be made for genome editing in farmed animals. I don't think most people see any fundamental ethical objection to the very idea of altering the genome of a farmed animal. I mean, some do, but I think most don't. But there are persistent concerns about animal welfare and naturalness that do need to be taken into considerations. I think narrow framings are likely to be inadequate. So anyone who's a proponent of genome editing in farmed animals, who just, for example, wants to talk about this marvelous tool and how they've begun to eliminate off-target events and have complete control over on-target events, that may end up, resulting in people saying, "So what? Why are you doing this?" Again, they want to see a focus on what are the potential benefits for animals and humans here, and how will it impact the system in the long term. They want to know why it's being used, but I think they will support some use cases. But the case will have to be made. Just a note on what's happening in the UK. I don't sense that marketing of precision bred animals in the UK is close. I am, as I said on my declaration slide, I chair the advisory committee, which advises the Food Standards Agency here on GM and also precision breeding. And whilst I can see precision bred crops on close horizon potentially being marketed, I don't see animals yet. I'm not going to dwell on why that might be. It's going to be a complex political question, and actually discussions between the UK and the EU over closer alignment may make the UK a market closer to the EU than we anticipated a few years ago. So, but I think at that point I will stop and invite questions subsequently. Thank you.
Thanks a lot, Andy. We'll keep questions for after Phil. It's easier then to kind of have all the questions together. Phil will now have the floor. Thank you very much, Phil, for being here. So Phil is from Wageningen University, and his expertise in science and society relations, but also with responsible innovation. He has led many different major initiatives, including the Just Editing project in the Netherlands, and will present insight on the public perspectives and governance relevant to gene editing in farmed animal. Thank you, Phil.
Great. Well, thank you very much. Is my slides being shared or not?
You have to share your slides if possible.
Okay, let's see. Right, how's that then?
You have to change the view. It's on
the view.
presentation, yeah.
Like that?
Perfect.
Perfect. Okay, great. Well, thank you very much for this kind invitation. And so my presentation, it fits very much with what Andy was saying, which was for me very, very interesting indeed. So I'm a social scientist. I'm a sociologist of science working in Wageningen. And I'm very interested, and this perhaps responds a little bit to what Andy was saying about technological exceptionalism. So I'm very interested in the democratization of science, so how to involve society at an early stage in technological innovations, including gene editing. And the reason that we're so interested in this is that science and innovation, it doesn't simply solve problems, it doesn't simply meet needs, it produces radical social transformations. And once you adopt that perspective, then there's really a need to instill sort of democracy or public engagement or different forms of involvement at the early stages. And so that's what's so wonderful about gene editing or GMOs. It offers the opportunity to experiment with ways of doing this. So this meets this fits within this wider framework, which is the theme of this, one of the aspects of the theme of this webinar around responsible innovation. And this is a framework which I was quite involved with, particularly in the UK, in terms of thinking about what responsibility could mean for science and innovation. And we focused very much on this as comprising four intersecting dimensions: anticipation, inclusion, reflexivity, and responsiveness. And a lot of my interest is to do with this sense of inclusion. So how do we anticipate societal concerns, societal responses? And this fits very much with what Andy was talking about in the Nuffield Council Bioethics report, which had this very innovative approach to public engagement. In fact, I was an advisor on that. And so this is the kind of approaches that we use. So we engage social groupings, sort of different kinds of publics. I tend to enjoy the focus group method as one way of structuring a kind of early engagement. And so the research that I'll be talking a little bit about really was trying to ask this question, and it was this: how, if at all, does the Dutch, so again North European, but this happened to be in the Netherlands, does the Dutch public imagine a future for gene editing in livestock, and if so, under what conditions? So there's quite a complex methodology, but as Andy said quite correctly, that it all depends on context. So we wanted to have discussions around animals in everyday life, around their views on livestock farming, and quite wide-ranging discussions before we introduced the notion of gene editing, both in general and how it can be applied to livestock. And we finished off with a discussion on governance. So there were different kinds of publics. We segmented them in these ways. There's lots of other ways to do them, but they were kind of public sector professionals, rural groups who lived in the countryside, people who were really into food and the outdoors, people who just had children, and people in the private sector. There's lots of other ways which you could segment it, but this was a sort of interesting segmentation that worked for this particular project. And so this was a collaborative project. It involved not just ourselves as social scientists, but also breeding partners, breeding companies. It was funded by the National Research Funding Body, and it involved also an ethics institute. So what do we find? And this, I think, is kind of parallel to what Andy was saying. So first of all, we found there was a great affinity with animals. We found people were very ambivalent to eating meat. And I think this goes back to what Andy was saying, is the difference between people as consumers and them as citizens, different sort of domains which they imagine themselves, their identities. Secondly, there was, and I think this is important in terms of the context, there was quite considerable and quite widespread sort of unease with the current livestock system. So, and I think this is a very much a parallel finding to what the public engagement exercise found in the UK. So within that, and I think this is interesting, gene editing was viewed as a techno fix. So it wasn't necessarily going to solve systemic societal problems, but it's going to have an attempt to fix at least some of them through technology. So again, there was this sense that gene editing under real world circumstances, and we can talk about that as well, there was this widespread perception that it would be likely to exacerbate. In fact, this is the word that Andy used. So it was likely to exacerbate inequalities due to the sense of what would happen if it fitted within our current market system. So that was one aspect that was very prominent. Another was the sense as to, well, why are we wanting to do this? Why do we think that we can improve on nature and naturalness? So this goes back to what Andy was saying. Not to say that everything is natural at the moment. Of course, people recognize it isn't, but this was a kind of an intensification of that move towards naturalness. And there was a worrying sense that this might inevitably lead to a striving for perfection. And that's a very, very interesting kind of argument. So another finding was that gene editing was seen as complicit with a hubristic attitude, a sense superiority of science to nature and its tacit ordering. So a sense that we can do the sort of the God trick and all that kind of stuff. And in relation to that, a sense, well, we need to exercise care, really deep care in developing responsible governance. So here's some indicative quotes, which are quite instructive. It's a plaster on a self-inflicted wound. It's a plaster on something we've created ourselves. So you might think that dehorning cows might have a welfare benefit, but is that really going to be the motivation which underpins them? You know, is it not going to be used just to further intensify the system? So, and this sense that there's a kind of transgression. Well, God has created the cow with horns. There must be a reason for it. Why do we think we can do better? Or is it going to be better only for the rich? Or is it going to be better for all of us in the world? And then the sense of, well, this will be just for those who have advantages anyway. Another sense about it being tampering with nature. So you might not know what the long-term effects are. So these wonderful, powerful techniques, which gives us lots of new possibilities to adjust, to change, to delete, to add the DNA of species. But the more powerful they are, the more likely, according to our participants, that there will be effects which we hadn't anticipated. And so to carry on, so we then looked at, well, perhaps there are some very deep-seated historical narratives which underpin public unease. So the narrative of the techno-fix, the narrative of the market rules, the narrative that we are wrongly seeking to perfect things rather than to leave things as they are, and that this constitutes a kind of messing with nature. So this is kind of very interesting for what constitutes good governance. And I think there's a number of prerequisites. So people don't simply just believe that you might imagine it's going to be a benefit for welfare, disease resistance, whatever else, even yield, but that's likely to be highly contested. So, and I think that's a very complicated discussion. Contested not least because what are the forces driving the innovation and what do we think about that? So what people had imagined would be a more adequate form of governance, which might not be totally dissimilar from the kinds of initiatives that Andy was involved with in the Nuffield Council. But it means asking different kinds of questions. So where are we going? Is this desirable? Is this really the kind of livestock system that we're aspiring to? Then who's going to gain, who loses, and by what kind of mechanisms? So I think these are some of the more sort of foundational questions about that. So to summarize, when we had this upstream debate, there was very little sense need for gene editing in livestock. There were strong latent public concerns about purpose, about trust, about whether we're included in the discussion, not just as consumers, but as citizens and early on, in terms of speed, as Andy has talked about already, and about equity. So really similar to what Andy was saying, there's very little fundamental sense that we shouldn't do that. But public acceptability or societal acceptability is conditional. So we need to move beyond simple evaluations of risks and benefits to the broader societal moral questioning of the technology. So that's important. So and actually that's a huge challenge really for what kind of methods that we can use for deriving that question. Gene editing seems likely to exacerbate systemic problems. So people need to be very much to convince otherwise. Underlying skepticism over claimed benefits, and that really covers most of the benefits we talked about today. And this sense of a need for more explicit virtuous, for virtues, not just in governance, but also in fundamental or applied research, whether that's in universities or knowledge institutes or in in corporations. So that broadly was what I wanted to say. I hope it's of some interest, and it's been a privilege to be part of this webinar and certainly to follow. And I think there's some interesting points of connection and perhaps some differences too. So thank you very much.
Thank you very much. Thank you, Simon, for being here. I know you have to leave. If there's any question, I will also bring it to you. Thanks, Phil, for your presentation. So before giving the floor to our speakers, which now is Andy and Phil, I would like to open this discussion with the roundtable. We have invited a few people to participate in this roundtable and to stay in this theme of society, ethics. I would like to, I see your camera on, Alexandra, perfect. I would like to start my first question related to this topic to Alexandra, who's from the University of Beirut and also works with the Project Detective, which is about traceability detection of NGT products, both in animal and plants. So Alexandra, my first question would be, from your perspective, what can already be done for research and innovation frameworks, even in the absence of clear regulatory pathways, because that's the problem right now with animals. So what do you think could be done?
Well, definitely try to push the idea that we need some regulatory changes, and the research has to continue. We see with the proposal now for NGTs that is concerning only plants, unfortunately, that the science and the research was also following the actions of the Commission. So I think it's very important that we have more and more data on the safety and also applicability of the technologies, so that we are capable of showcasing, if there is any regulation to be proposed by the Commission, the more data it has, the better and the easier in the end. Also, as we see with the NGT proposal now, the trialogue takes a lot of time, and it's a very, very political kind of a debate among the member states, but also between the different institutions within the EU. So I think it's important to be prepared from the scientific point of view, so that the discussion, the political discussion, can also refer to some scientific arguments.
Thanks, Alexandra. I have a follow-up question on this. So based on everything you learned today and the work you have been doing in Detective, what do you think we should do in the animal sector in terms of detection, traceability, and identification? Because as you saw the different speakers earlier, they showed also how selective breeding is very similar to the concept of gene editing. So what is your legal expertise on this and the work you've been doing at Detective?
I'm not a scientist. I'm just a lawyer. But as we have already discussed within the project, but also it's also seen in the work that we have been doing and in the publications that have been done during the project. So now it seems like there is no leeway from what we have in terms of the current legislation if we talk about the identification methods, which have to be analytical, PCR based, most likely. That's also very difficult to sometimes state what the legislation aimed at. However, we are sure that they have to be analytical, they have to be even specific. So not only we have to be able to detect the changes within the genome, but also we have to be able to identify what caused this genomic change. So I don't really think that scientifically speaking, there is so much to be done to somehow be compliant with the current legislation. Within the proposal, the idea is to relax these measures and for the NGT1 plants not to have any kind of identification methods which are analytical. And for NGT2s, there are methods foreseen that are even specific and are capable of identifying. However, there are also certain leeways not to provide them if it's not technically possible. So unfortunately for now, legally speaking, there is not much to be done until the legislation is not relaxed, especially if we talk about the very small changes in the genome that are mimicking or very, very similar to classical breeding techniques or changes that are basically occurring in the nature. So we can only hope for legislative changes, which in plants after December, the hopes are high. I would say that the proposal is going to be actually adopted. We'll see. Now it's on the table of the council and the parliament, but we are getting closer. And with the study on NGT animals from EFSA, there are also some hopes that a proposal for NGT animals will be prepared by the Commission, basically.
Thank you very much, Alexandra. So seeing this as a technical bottleneck, now I would like to invite three people from the breeding sector, which is who are Clotilde Patry, Ashley Norris, and Clint Nesbitt from the different sectors, so aquaculture, ruminant, and swine. So thinking about this technical bottleneck, which is the regulatory approach, what do you think is still limiting in the sector in translating the gene editing from research into breeding and production systems based on the different presentation, but also based on what Alexandra has said? So I don't know, I think I see Clint, if you want to take the floor first, and then we can move to Clotilde and Ashley afterwards.
Yeah, thank you. I'd be happy to. Thank you for having me here this afternoon. So I'm Clint Nesbitt. I'm with Genus PIC. We're the British breeding company that's trying to bring our gene-edited PRRS-resistant pigs to market. And I think for us, really the limitation is less about the science and more about some of the broader issues that we've been discussing. The need to have a functioning regulatory system to ensure the safety of the products and to be able to even approve them. Many countries in the world don't even have a system at all. So certainly the regulatory hurdle is very significant. But then I think the second hurdle is very much market acceptance and understanding how the public will perceive and accept the technologies when we do begin to bring them to market. I think because of both of those challenges, unfortunately, in the near term, we are going to see this bottleneck where really only large companies with very high value traits are going to be able to have sort of economic justification to even attempt to bring things to the regulatory system, and then still have to face all those uncertainties associated with consumer perceptions, market acceptability, and the like. We've conducted a lot of research ourselves on market access, and I think are seeing things that are very similar to what Phil and others have talked about. Generally speaking, it seems that there is more public willingness to consider traits that do genuinely improve animal welfare and animal health. So that makes us a little bit more optimistic about disease resistance. But we do see similar patterns where there is questioning about the utility and wisdom of using gene editing tools for things like mere production traits, sort of the way it seems to be perceived, yield improvements and things like that. We also are paradoxically seeing in our research that consumers may be less willing to adopt things that are perceived as being directly and solely for consumers, which seems a little strange to think about. But things like meat quality, fat content, color, mouthfeel, those sorts of things, I think often get less support. But just because I think they're perceived as being of benefit only to humans in sort of a utilitarian sense. So I think if anything, just to circle back, in the near future, it's going to be tough to bring some of these traits to market. But unfortunately, I think probably the bottleneck, the limiting factor, will keep it to likely large traits with fairly high value benefit that you can sort of sustain, making it all the way through the regulatory system to even get to that point of testing to see what market acceptance will actually look like.
Could I just make a very swift response to that, which might be very... So I completely agree with you, but I'm very interested in terms of the way you talk about consumers might be more skeptical or less positive about innovations which are meant to help consumers. And I think that's a fascinating sort of paradox in a way. But then also what underpins that really is, well, what do consumers, or what do citizens want? And so there might be these promises about color and texture and meat, whatever else. And, is this really what people want? So that's one question. People might be already quite ambivalent. So why should we, and the other one is to say, and I think this is really interesting, is that, well, if you're going to justify such a radical intervention like gene editing, which is a radical intervention into DNA, then you're going to have a bloody good reason for that. And these are not good reasons. So in a sense, that might help explain a little bit that paradox. So you're going to have to talk with them as to what is a good reason that would warrant this intervention.
And that is absolutely right. I think you've hit the nail on the head. That really is, I think, where the perception is coming from. That needs to be that justification that's acceptable for why you're using this particular intervention. Yeah, absolutely.
Yeah, but that's what's fascinating, because regulators don't tend to regulate justification or good and bad uses. They regulate safety and risk. But there are some exceptions there, like the Swiss are experimenting in relation to their new gene editing frameworks, to whether they should include a benefit evaluation. So what would constitute a benefit? And again, that opens questions as to who's going to assess it, what's going to merit a good benefit or not. But at least there's a move in that direction. And, the sort of Norwegians also moved a little bit there. And I think that's really interesting, because at least it starts from the point that people are not just concerned about safety. So regulation of safety is necessary, but it's not sufficient. And that's what goes back to the sense as to what are these innovations likely to be part of? And is this really the kind of world social relations environment that we're after?
Thank you very much. Maybe it's good, because we had the impression of Phil, which gave a bit of the, and Andy who gave a bit of the UK perspective, and Clint on FDA approved in the US. So Clotilde, I know in France it's a bit more complicated, especially in ruminants. So would you like to add something here?
Yes, so thank you, Gina, for inviting me to participate to this roundtable, and good afternoon to everybody. And thank you to all the speakers. So it was quite interesting to see all these advances anyway from a scientific point of view. What I could add is, so there are still some technical or scientific questions to be solved, of course, because first, if I'm talking about ruminants and especially cattle, when we are discussing about gene editing, our focus would be more on adaptation and disease resistance. But in fact, we still lack knowledge to really work on adaptation and disease resistance. And this is also the question of the heritability of the trait, and it has been mentioned in one of the talk. But this is first one part. The second part is maybe the adoption by farmers and the acceptance by farmers, because this kind of discussion, I think, thanks to IFAB, for example, and all the projects that are engaged at the European level, there is a lot of discussion and new knowledge about genome editing, but we still need to discuss it not only at the research level, not only at the consumer level, but also breeding companies and also breeders, farmers. They need to be aware of this kind of technology and also to accept it. And I think there will be a big question of costs, because for sure, we can be afraid of the increase of the semen cost, for example, due to this kind of technologies. And this will be a big barrier, I would say. And the last point, to be short, I think one of the speakers has mentioned that the most important is also to be aligned on what are the main goals for the future of agriculture. And maybe this is not always how to reach these goals, but what are the goals exactly? And if I'm talking a little bit about the ruminants, so we have this will to be able to adapt to a changing environment and to be also quite autonomous. For example, this is true for feed and food, but I think the question can be raised also how to be autonomous in our practices and maintain our ability to adapt while we don't know exactly what will be the future. And when we start talking about biotechnologies, at the same point, we can go faster, but we need to have still flexibility. And that's why sometimes it can be discussed and it can be opposite to our will to be autonomous and adapt and to maintain our capacity to adapt. So I think that's the 3 points that I wanted to mention, and I'm sure that we could discuss even more on it.
Thanks a lot, Clotilde. I think there's a question for Andy and Phil in the chat box. So it's from Xavier. Thank you for these excellent talks. Question to Andy and Phil. Do you estimate that some stakeholders are still missing at the regulatory table discussing animal breeding and, of course, precision breeding in general? And if so, would you have any suggestions to have them included better?
Phil, why don't you have a go at that one?
Well, thank you. Yeah, perfect. Well, there was a very interesting experiment. Andy might remember it. About 20 years ago, there was something called the AEBC, the Agriculture, Environment, Biotechnology Commission, which didn't last very long, but it was set up in about 2002 and it survived until about 2006. And that was fascinating because there'd been such a polemic, but a short time before, it really had a very broad range of stakeholders at that commission. There were NGOs, there were consumer bodies, there was Gene Watch, then there was a range of different kinds of academics. And it as Andy probably had with his experience in the bioethics thing, it makes for very difficult conversations. But it actually led to some brilliant reports out of that. So I do think that there are lots of stakeholders which are not part of their discussion, principally because regulation is usually narrowly defined around, technical risk, whereas the issues are much broader. And once you then follow that through and say, if they're broader, then different kinds of stakeholders should be at that table, from civil society, from different public groups and whatever else, but also with different disciplinary competences. So, to involve whether it's political scientists or sociologists, anthropologists, theologians, ethicists. So I do think that it's absolutely critical that we do broaden the base of the kinds of stakeholders that are involved in these processes. And we did some research comparing responses between Brazil, Mexico, and India and GMOs. And these regulatory bodies were a nightmare because there were all sorts of issues surrounding GMOs and their use in agriculture and the intensification of agricultural processes, their impacts on peasant farming, whatever else. But there was no space for for these issues to be discussed, apart from in these very restricted regulatory settings, which then became a nightmare for everybody involved. So I do think we really need to, use our imagination to construct regulatory arenas that are kind of fit for purpose. So that would be my response to that.
If I could just add to that relatively briefly, I mean, I suppose without wanting to sound flippant, the main stakeholder that is missing from the table is the animal, of course. Now, I know there are some people who there are initiatives about, animal. There's one called Animal in the Room, I think. But of course, practically, there wouldn't be much use to have a ruminant in the room as a regulator is considering an application. So, putting that to one side, I think probably I think that the questions about roughly and broadly the kinds of interventions that we support as a society are political decisions. And I'm not entirely sure that they do belong at the regulators' table. I mean, I think the regulators should be given direction by whatever government or democratic institutions are in control in that jurisdiction. They should be given general, broad guidance about the kinds of interventions that are acceptable, and then they make their decisions on rather more narrow criteria. Because otherwise, I think it'd be very difficult to operationalize some of the kind of issues that Phil and I have been discussing. If every single time we have to make a decision about an application, they have to be discussed from fundamental ethical principles upwards. So let's have broad political guidance and then detailed regulatory decision making, but certainly that regulatory decision making should involve considerations of benefit as well as risk, and should involve opportunity cost considerations and relatively broad policy context. I think that's often what's lacking is a broader policy context. But yeah, it's a really interesting conversation. As someone who works with regulators, I should add that they are all overworked and understaffed and under-resourced. Right. So if you want regulators to be slick, you better put your money into them.
Thank you, Andy and Phil. Talking about the animal in the room, I think I want to go back to Clotilde and Clint. I don't know, Ashley, if you're still here, but to address the question raised by Dr. Jackson. So you work a lot with farmers that are your main client, and that's who we want to target at the end of the day. So how do you address the possible concern about unintended consequences or long-term impact of gene editing? Because again, at the end of the day, the farmers are the one receiving the products and then going onto the market. And we have two different perspectives. We have the perspective of Clint, who has the FDA approved now, and then Clotilde, where the process is still far. But how do you think we should address those possible concerns?
Well, certainly, I think that in our case, the way that it works is that the regulators themselves address their needs to assess whether there is the likelihood of long-term effects that should be of concern in their initial regulatory approval process. And then if there are any sort of lingering doubts about what could come up in the future, they will sort of impose upon us additional sort of longer-term monitoring plans such that we're required to watch for certain kinds of things. And if those things come up, we're required to notify the regulatory agencies that something has changed. We will also certainly include contractual language with our buyers, our customers, that they are to report to us if they are to observe certain things that are appearing that we didn't notice before for some reason. So that's sort of how we're handling it in this particular case. In most markets, it is a single regulatory approval by a government agency, and then sort of pairing that with longer term observations over time, just in case.
Thank you, Clint. Clotilde, do you have?
not so easy to answer the question, but it has been mentioned that we need to collect more and more data to showcase and to bring some scientific proof. And I would join what has been said just before. If so, people, they always need to anyway to experience this new things and new approach. And for sure, if we cannot anticipate any I would say negative phenotype or unexpected phenotypes, for example. But it's very important, like for any genetic abnormality, to collect the information and to be able to share then this kind of observation and then to prevent before any, I would say, propagation of this kind of side effect. So, but it could be a kind of participatory approach. And in that case, everybody would be in the loop, I would say. So, but as I said at the beginning, quite hard to answer.
Thank you, Clotilde. Thank you, Clint. Clotilde, you mentioned data. I would like to bring this conversation back to Eurofaang and give the floor to Emilie and Elisabetta now. So, Emilie, Elisabetta, how critical do you think that in vitro and cellular models, shared data standards, and research infrastructure are responsible for accelerating responsible development in gene-edited traits? I don't know who wants Emilie, yeah, or Elisabetta.
Elisabetta, you go first.
I go first? Oh,
yeah.
Okay. So, well, recently, in this recent year, I become interested with in vitro systems. And I think that in this context, this is a wonderful possibility to reduce the use of animals, or at least to focus in an efficient way on the changes that really need to be done on animals in a proficus way, because you can test in vitro a lot of maybe possible undesirable effects, at least at the cellular level. You can really prioritize what changes you'd like to do. And that is the main thing, I think. It's something which is kind of easy, if you want to do, because now you have several systems that are extremely complex, more physiologically relevant versus the physiology in vivo. And this doesn't replace at all the living animal. This is quite clear to everybody, not maybe to some parts of society, unfortunately, but I think scientists know this very well. But knowing this and mimicking as much as possible the reality of in vivo, I think you can make more effective the process of genome editing, which is really worth. Yeah.
Thank you, Elisabetta. Emilie?
Yeah, so linking to what Elisabetta was saying, so the data infrastructure, it can provide tools and ways to store information, particularly from, for example, CRISPR screens to identify new targets, information about multiplexed edits, and make that information more accessible and fair to the research community, so that the access to those tools and resources to be able to do those CRISPR screens, for example, is more equitable across Europe. Data infrastructure is also important, I think, in terms of characterizing the animals, so phenotyping the animals, making that data available, particularly in the kinds of experiments that Anna was talking about, where they're phenotyping edited. They could be phenotyping edited animals at scale. So the data infrastructure can certainly help significantly in driving the space forwards, for sure.
Thank you, Emilie. So in general, I would like to kind of close up soon because we're already over time. But before closing, I would like to ask each of our speakers and roundtable participants, if Europe were to prioritize concrete actions over the next 5-10 years to really enable responsible and impactful gene editing in farm animal, what should they be? So I would like to start with Alexandra in terms of legal perspective. What do you think it should be?
Well, starting to think about the way we want to regulate it. I think that now with the plant proposal, NGT plant proposal, the animal one would look quite similar. And if it works, then I guess it's gonna open certain doors. Although let's see if politically it will be as easy to push forward. But yeah, basically, I think that it's important now to just focus on all of the things that have been said today, maybe also on the more ethical aspects of regulating the technologies. Although I'm quite afraid that in the setup that we have at the moment, it might be quite difficult to do so. But yeah, I think that for now with the NGT plans proposal, we just need to wait and see how it's going to evolve, and it's going to then immensely impact the future for the regulation.
Thank you, Alexandra. Andy, what is your concrete action you think for Europe?
Yeah, so interesting just to hear about precision bred plants because we haven't really discussed them today because of course that's not our subject. But I do think it's instructive a comparison between the debates and discussions we've had around crops and precision breeding versus animals because some of the objections in animals are not easy to sustain in plants. So, for example, the objection about naturalness. The problem with plants is that our traditional breeding programs are full of unnaturalness. We bombard them with chemical mutagens and X-rays, and that seems to be acknowledged as being part of traditional processes. So I think continued public engagement, continued public dialogue, continued stakeholder engagement, having these really hard conversations, which sometimes are hard and uncomfortable, but continuing to have them so that we become, so that whole debate becomes normalized. It's ongoing. It's not just for Christmas. We're having it the whole time. And then I think we grow up as a society so that we can make tough policy decisions because this will be a tough policy decision. And there will be people who strongly object to the first genome edited animal that's marketed. But I think it will be at some point.
Thank you very much.
Just one follow-up question because I'm quite curious about this because my impression is whenever I see people and talk to people and we were also conducting certain citizens juries and all of these kinds of science communication with regard to GMOs and gene editing. And I have the perception that, as you said, of course, with animals, the discussion about naturalness is a bit different and shaped differently because of the fact that in animals, we have mutagenesis, random mutagenesis, so chemicals and radiation. But I wonder if you really see the difference in the perception of the consumers in this regard, because I have the feeling that there is still a lot of misunderstanding among the consumers with regard to that. I have the feeling, and it's a very anecdotal evidence because I'm not specifically working on the consumer perception, but I have the feeling that people are not very aware that they are actually consuming GMOs. Meaning random mutagenesis products. So that's also another layer of the discussion that is quite difficult, I think, in terms of science communication and finding ways to regulate it. Then
I'm sure there are lots of I mean, science is hard, right? Social science is hard, and there's lots of misunderstandings when it comes to any kind of theory. I'm sure Phil could talk to the cows come home, metaphorically speaking, that is, not the genome edited cows. Phil could talk to the cow comes home about the so-called deficit model, where many scientists have assumed that if you just teach publics enough about science, they'll agree with you. And of course, that isn't the case. So we are still talking about kind of the values that are at play when people object. I think I do think there are inconsistencies, however. I mean, just because the deficit model is wrong, that doesn't mean we're a scientifically literate society. And there are misunderstandings about what is natural and what isn't, and the normative dimensions of naturalness. I can't think of many other more natural diseases than cancer, because it usually gets all of us by a certain age. Perfectly natural, but perfectly objectionable. So, I think sentience is the key thing. Animals are sentient, and that matters to people.
Thank you, Andy. Phil, what is your concrete action for Europe?
Yeah, sure. So my concrete action is to open a debate, a conversation, a hard, ongoing conversation about what constitutes a benefit, which is a benefit that's kind of co-produced with and for society. and then to develop experiments on how to institutionalize that in terms of policy architectures. So that's that. Just on Nandi, I have to comment that. So I really like that sense about naturalness and such like. But we did some work on public engagement on and gene editing in plants. And we actually focused a lot on mutagenesis as well. And there was a lot of, when people actually, aim to be aware about what happens to mutagenesis, they weren't happy with that at all. So, so that was very kind of interesting. I mean, that's part of these sort of systemic problems. And so how to make a more inclusive conversation would be really important about that.
Yeah.
Thank you. So, Clint, for you, it's even more, you need even more concrete action, I suppose, because you're based in the US, but a lot of the company part is in the UK and in Europe. So for you, what would be a concrete action over the next 5 to 10 years? And I hope you will say, no, you need it next year.
Well, at the risk of completely repeating the other panelists, I think obviously the two priorities are we need a functional regulatory system in the EU and the UK to assess the risks of these products. But I think simultaneously, we also need to really reinvigorate or really lean in on the societal engagement and really having that conversation about how consumers and society feel about the use of these technologies in agriculture.
Thank you, Clint. Clotilde?
Sorry. I would say that maybe we should work on the question in between genome editing and animal welfare. So because we always said that animal welfare is much more complex than, for example, the effect of gene editing, but still this is a regular question, and we saw it in the EFSA risk report, and that's why maybe we should work on it. So in order to bring a few answers, and maybe another action, maybe quite concrete and not only for Europe, but maybe to introduce in the education program from school. I mean, it could be in agricultural school or even lower level, but this notion of what is genome editing and what could be the potential of it. So, but to discuss it early in the age.
Thank you very much, Clotilde. That's a really good point. Elisabetta, please.
Oh, I don't know what will be exactly the priorities in the next 5-10 years, apart, of course, trying to be as focused as much as possible on the mutations to prioritize. One thought I had during this beautiful event that you organized, Gina, was that maybe more far in the future, I mean, imagine near future, you have animals that are edited for some number of edits. I heard up to 13 for this wolf that was shown earlier on. And these animals will still undergo selective breeding. At least this is something that companies do. And then on the same genomes, we will add new editing probably. No? So it will be a cumulative process. And we don't really think so much about that for the moment because it's quite far away. But should we simply accept that somehow naturalness is lost? I mean, we should assume it somehow. Or maybe we should also assume, okay, we make a big change as it was already made by other means in the past, because even selective breeding, traditional selection is also a big thing that we do to naturalness. We have always done. Maybe in this case, it is nature which adjusts the consequences and we can be confident of that. In the other case, it will be us to face the consequences. But I think it's something that could be quite fearful for people who may prefer to go slow versus speed, So it's a philosophical thinking that I had on top of all.
Thank you very much, Elisabetta. So, Emily, I would like to give you the last bit and also the message to wrap up as the co-coordinator of Eurofaang. What is your concrete action?
A concrete action. So just to wrap up, saying that it was really useful to hear about all of these talks, the tool developments, applications, the regulatory changes, and also the socioeconomic concerns. And it's just such a rapidly evolving space. Since we started Eurofaang in 2023, things have changed massively. And the acceleration in this space is huge, I think. And the changes around, particularly the discussions and what we're discussing are really exciting. Obviously, there's woolly mice and colossal and all those exciting things, and multiplex editing on the horizon, and the infrastructure really has to be developed to go along with that. And I think that's what Eurofaang really showed, is a consolidated research infrastructure in Europe that links stakeholders, builds capacity and increases equitability across Europe can only be a good thing and can only drive progress forward. So obviously we hope GenoPhoenix proposal will be successful, but then also we hope that the foundation provided by Eurofaang will, continue to grow and continue to bring people together so that we can continue in this space. And how we do that is sort of an open question at the moment. So we'd be certainly glad to hear feedback from participants in the think tank about where we go from here. And thank you very much, everybody, for participation today.
Thanks,
Great.
Emily. Thanks very much. I don't know if we have questions in the audience. I don't see any hands up. But thanks, everyone, for attending today, for your participation in the roundtable. I think it has been very effective, and we had a lot of important discussion, and people still being in the room after 23 minutes that the webinar is supposed to be ending shows that there's a big need in the conversation, that there's a continuity, and that right now it's becoming more and more important, not only the technical aspect, but also the social ethical aspect of gene editing, and that it carries such a weight in the conversation today. I would like to let you know that we will provide this webinar online, and I hope that you all really enjoy this interesting talk, and for everyone who made the time, I thank you once again. I know we're in different time zone, but we will keep you updated. There will be a lot of information still on the Eurofaang website, and then slowly fading into the GenoPhoenix network. So thanks a lot, and hoping to see you again soon.
Thanks, everyone.
Thank you a lot.
Thank you.
Thank you very
Thanks,
much.
Gina. Thanks, everyone.
Bye-bye.